I'll talk about the microbial world and trying to shine light on what I might refer to as dark matter. So historically, microbiologists have really been restricted to looking to that part of the microbial world that we could readily culture and study in the laboratory. But because of various methodologies, we knew that this was really only the tip of the iceberg, that the great proportion of microbes in nature, including in medical scenarios, are not yet culturable or have been resistant to culture. And so I guess collectively, I'm referring to that as microbial dark matter. It's the majority of the microbial world that sits in this region that we know it's there, we know it has a role to play, but because we have either not yet cultured it or it's resistant to culture under the conditions we use in various practices, it sits there undetectable or as dark matter. And the genome sequencing technologies, I think, have provided us new opportunities and applications to facilitate looking at the microbial world in a holistic and a mechanistic way, because it allows us to be independent of cultivation. So as long as we can recover a precise and accurate representation of the microbiology from the sample we want to study, and we can facilitate a non-biased recovery or extraction of that nucleic acid, these sequencing technologies, when applied to that particular template, are starting to give us an insight into both the structure of microbial communities in terms of what types and kinds of microbes are there, as well as the functional characteristics. In other words, giving us a blueprint or a wiring diagram of how these microorganisms actually work within the system. So it's opened up opportunities to look at more complex and diverse mixtures. And I think the costs and the time associated with the advances in the technologies have also given us opportunities now to work with not just real-world samples, but also work in a much faster and comprehensive way than we've ever been able to do before. So the quantity of samples from single individuals over a longitudinal fashion, as well as in a cross-sectional or observational way, the quantum of samples that can be analysed and the holistic information that can be produced when combined with the power of computation and statistics is giving us some real insights in terms of what's going on. So how do we do this? So principally, if I look at the microbial community, really the key step in the initial phases is making sure, as I said, that you recover this in a holistic and a non-biased way. And so groups like mine have spent a lot of effort in terms of trying to optimise and get those conditions correct in terms of making sure that the community is sampled in a holistic and a non-biased way. And then the next step is to make sure that the extraction of DNA from all of these different types of cells, which vary in terms of their structure and rigidity or resistance to breakage, is effectively achieved in a way that all of the DNA or the metagenome from this collection of microorganisms is released. And then the DNA can be used in two different ways shown in this panel here. So the first one still is based on a PCR-based approach, which takes the DNA and uses it as a template with PCR primers that amplify regions within a specific gene that is universally conserved across all cells, not just prokaryote, but eukaryote cells as well. And that's the gene that encodes for the RNA that is present within the small subunit of the ribosome. So this is about 1,800 nucleotides in length, this gene, in the prokaryote world, and we can amplify specific regions of that from the metagenomic sample. And the colour code here is to give you an indication of the different sequences or amplicons that are produced. And these can then be compared to databases or dictionaries, if you will, in terms of other sequences that have already been collected and deposited and described or characterised in a taxonomic way. In other words, we've got an idea what specific microorganism that relates to. And depending on the sequence similarities, we can start to bin the sequences into what we might call operational taxonomic units, or OTUs. And basically this is building subpopulations out of the total community that gives you a representation of not just who's there, but the relative abundances of these individual taxonomic groupings. And then using statistical analyses, you can start to look at variations both within the complexity of the sample, how many different types and kinds of microorganisms may physically be present in that sample, as well as the relative abundances of those. And then statistically, these can be shown in covariate space and give you an indication of how similar or different these populations are with respect to each other. More recently, the DNA sequencing technologies have allowed us to kind of circumvent the PCR step and go ahead and make libraries of DNA and just go ahead with what might be called shotgun metagenomic sequencing. And in this instance, what we're doing is producing sequence data for genomic information in toto from that microbial community. And again, using dictionaries or databases, trying to align those sequences with something that is currently in databases, either provided by reference genome sequences or by other sources. And while that dictionary isn't quite as good as the one we have for 16S ribosomal RNA genes, if we think of that as Webster's dictionary, I think the functional dictionary is more like a Macquarie dictionary. I think we're getting to a point where we can start to look at the compositional or functional attributes of the microbial community. So I guess the point I'm trying to make is sequencing technologies have opened up the microbial world to being examined and characterised in a more holistic and mechanistic way than what we could previously achieve just with culture. And as we move forward with advances in the technologies, the capacity to use these with real-world samples in areas of basic research, in high-quality, well-designed clinical trials is enhanced. And in addition to that, the opportunities potentially to develop new diagnostic tools that may be useful in clinical scenarios or to change clinical practice could be an outcome of the application of these technologies. And so with the rest of the time I have, I'll give you just two examples of some collaborative studies that we've started here since I moved to Diamantina Institute to try and give you an overview of how we're trying to contribute in this space. So the first example I want to talk about is a collaboration with the Centre for Liver Disease Research here at PA Hospital, led by Elizabeth Powell. And basically what we've been looking at is the microbiota composition, both in terms of a structural and a functional context, that can be found resident within Ascites fluid. And the first paper arising from this work was recently published. And as the title suggests, what these cultivation-independent techniques have allowed us to do is get a quantification of the bacterial burden of Ascites fluid in different patients and how an increase in that bacterial burden enumerated without the need of culturing organisms using these sequencing technologies as a tool has been associated with poorer clinical outcomes. And this is a summarisation of the data, and principally you can see in each of these figures here there is a positive correlation that's statistically significant with respect to the number of neutrophils in Ascites fluid compared to the number of bacteria also identified present there. The albumin gradient also appears to impact the burden or the quantity of bacterial cells that are present in Ascites fluid. The impact of antibiotics seems to be a minimal change or driver in terms of the relative abundance of microorganisms, at least suggesting in these 25 patients that have been sampled here at PA Hospital that there may be a level of antibiotic resistance genes resident within these communities that don't really knock down the population to any significant extent. But I guess more importantly, even in this pilot study, an increased bacterial load, which wouldn't be detected necessarily or easily by culture-dependent techniques, has been associated with a significantly increased mortality rate. And the time to readmission for these patients all suffering from decompensated cirrhosis is shortened depending on how many bugs are actually found in the ascites fluid. So I guess this is one area of basic research where we've taken a cultivation-independent way and have just looked at bacterial load. At this point, we're still working through the data sets with respect to looking at the types and kinds of microorganisms that are in the ascites fluids captured from these patients. And in the future, we want to also use these techniques to look at the array or the spectrum of antibiotic resistance genes that might also be present in these communities. But I think that this is an important application of what is a relatively rapid, cultivation-independent technique that suggests that it provides an important insight in terms of risk management and clinical management of patients suffering from severe liver disease. The second example I want to talk about is collaborative work really done by a NHMRC clinical research fellow, Li Zhang, who's based, or her home department is with Claire Rickard's group at Griffith University. And we've been supporting her work to look at the bacterial diversity associated with indwelling catheters in patients that are ultimately admitted to intensive care units. And so basically what Lee has been interested in learning more about is the extent and the microbial diversity that forms a biofilm, a living surface, so to speak, that colonises and is retained in these various catheters, and what types and kinds of microorganisms are present there, and also learning something about the functional attributes of these microorganisms. So I'm sure there's more people here with much more experience and knowledge than me in terms of bacterial sepsis, but at least in terms of cultivation-based diagnostics, there's a very low success rate, as I understand it, and to a certain extent, even PCR-based diagnostics is an improvement, but it's not a 100% success rate, either in terms of identifying the pathogen involved or giving you an indication of what that pathogen actually is. So there are issues associated with the management of patients and the use of things like catheters, where these cultivation-independent techniques can provide insights in terms of what's going on over time and may have a potential influence on nursing and clinical management of these patients. So in this particular study, there were 18 subjects. Most of them, virtually all, were male. The key notable features are listed here at the bottom of the table, but you can see their ICU stay ranged from 1 week to almost a month. And basically, all of these persons had indwelling catheters for the period of time that they were in ICU, and most of them received a broad range of pretty potent and broad spectrum antibiotics during their hospital stay. And this is what was found once these patients had the catheters removed. There was work done looking at taking the tips from some of the catheters and doing cultivation-independent sorts of assessments in terms of what sorts of microbes might be associated with the catheter. And then we used the 16S-based approach to try and characterise the community in a cultivation-independent way. And I think the key point I want to make here is that the variation, both in terms of the success rate associated with cultivation and what was being recovered and identified by those cultivation-based methods, is very different to what we saw with respect to the cultivation-independent or 16S ribosomal RNA-based approaches. And in particular, this bacterium, at least with the catheters used in this particular study, recovered from the patients here around the Greater Brisbane area, this bacterium, Ralstonia piquetii, was present in very high abundance within these biofilms that formed in both the arterial as well as the central venous dwelling catheters. So we actually went ahead and isolated some of these organisms, in particular Ralstonia, and we went further in terms of producing genome sequence data to give us an indication in terms of what functional attributes that microorganism had. So basically from a pure culture, we extracted DNA, ran it through a library construction to use on some of the next generation sequencing technologies. These are just indications of the depth of coverage in terms of the number of reads over the region of assembly, and ultimately we were able to bring this together into a complete genome. And the colour coding dashes here are an indication of our capacity to look at dictionaries and make an assessment with a degree of confidence about what particular gene is encoding what particular function. And what we learned from this particular piece of work was that the Ralstonia picketii strain that Lee isolated, in addition to this bacterium being in high relative abundance in the biofilms of the catheters of the patients, the isolate that she recovered had some features that would suggest that it's multiple drug resistant. So you can see the number of genes involved with efflux pumps. So this isn't necessarily a modification of the antibiotic, but the microorganism escapes being affected by it because as soon as this enters the cell, it's actively pumped back out again. So there is no effective build-up or accumulation of the antibiotic as a result. And in addition to those efflux pumps, there's a relatively large number of beta-lactamase genes and a variety of other genetic or coding sequences that we could predict would have a role to play in antibiotic resistance, or even more generally in multiple drug resistances. So we identified this organism really initially through a cultivation-independent way, and then we used techniques inherent to the specialist skills and capabilities that we have in my own lab group to facilitate the isolation of this organism. And then again, by using these genome sequencing technologies, started to develop a picture that this organism is a substantial reservoir of genetic information that encodes for resistance to multiple types of drugs. So I think to wrap up, I think, at this stage, I hope I've given you just a couple of examples of collaborative work. This isn't a major focus of our research program, but in collaboration with people like Elizabeth and Kate Irvin in the liver group, as well as Claire Rickard and Lee Zhang in terms of nursing and clinical practice, I've given you a couple of examples where these cultivation-independent techniques and the sequencing technologies give us insights into the microbial world in a much more holistic and mechanistic way than we had before. And at least in some cases, it's going to be the applications of these technologies looking at various sorts of studies in the clinical setting that may provide information that is non-biased and representative of what's happening in the real world and may inform and change clinical practice. So the next step in Lee Zhang's work as an NHMRC clinical fellow is also to start looking at connectors and seeing if there are variations within connectors with respect to the development of these biofilms and whether they actually physically serve as a reservoir for these potentially infectious organisms. And of course, the idea, and many people hold great excitement in the application of some of these new technologies like the nanopore sequencing that Matt introduced you to this afternoon, as very rapid tools for the diagnosis, identification of pathogenic microorganisms, like in intensive care units, bring that identification down from days to a matter of 1 or 2 hours, with the view that a much more informed or powerful assessment or determination could potentially be made in terms of which antibiotics to use under what scenario with each individual patient. So just to finish up, I just want to acknowledge the collaborators, and some of this work was done while I was still at CSIRO, but thanks for your attention.